Hello, everybody, and very welcome to this research seminar from this Genomics England Research Seminar. Today, we're going to be looking at research which is showcasing research from the population genomics community. I am Eoin Scally, so I am one of the co-leads of this community, along with Hilary Martin, who's also going to host this seminar today. The seminar will last about an hour, I think, and it will be recorded, and I think will then subsequently feature on YouTube, so we can all review that there. And there's a couple of points I think I'm supposed to tell you about how to contribute to the how to ask questions. Let me just bring up these notes. Yes, so there'll be a Q&A after each speaker. So we'll have two speakers, and you are asked to use the designated Q&A feature rather than the general chat to ask questions. And that hopefully should be clear when people are using it. Okay, so this is the first research seminar from the population genomics community. So I'll just tell you a few things about the community itself. Let me just move this out of the way. So here we are in relation to the communities in general. Population genomics, I think, was one of the communities that has survived from the changeover. It was formerly a GSIP, established around at the start of the 100,000 Genomes project. And its aims are quite broad. It's kind of collects together projects and researchers interested in exploring and using the Genomics England data to address sort of population scale issues, population health, obviously, and disease, but also more fundamental research, sometimes into kind of processes that shape the genetic variation that's there. So evolutionary and demographic processes involved. And so we've got about 378 members. I'm sure that's accurate. And we have I think we're about sort of in the middle of the group, so it's a quite a diverse group. It's one of the more kind of interdisciplinary groups of the various communities. This is the current leadership team. There's me at the top. I won't go through everyone's affiliations and research interests, but you can see them there, and Hilary at the bottom. And we've got two others, Patricia and Jean-Baptiste, with kind of spanning these various aspects of research into human genetics and genomics. And the aims broadly of our aims, I would say, as a team trying to sort of develop this community, are to identify opportunities to build collaborations and networks, and that we're hoping that it can be a useful tool for that, and as well as facilitating sort of research into the GEL data using from this perspective. And I think that's a sort of an ongoing project for us to sort of work out best ways to do that. So I have a couple of announcements that you can see here. I can see Genomics England will be at ESHG, which I think is in Milan. Which sounds very nice. That's next week. So if you're there, you'll see them. And also then there's the Genomics England Research Summit next month, which is always a very successful meeting. Encourage people to go along to that down in London. And you can use these QR codes to register. Okay, so I will pass on to hand over to Hilary to introduce today's speakers. And I think we'll have Isaac first. And then Luis. So Hilary.
Hello, nice to meet you all virtually. So the first speaker is Isaac Garcia Salinas, who's actually been a...
Sorry, Hilary. It's gonna be Luis first because he needs to leave earlier. Sorry.
Oh, I see. Okay, it's gonna be Luis. Right. So Luis is a clinical academic cardiologist, consultant cardiologist, focused on cardiomyopathies at Barts Hospital, and also associate professor at UCL in the Center for Heart Muscle Diseases. So introducing Luis is going to tell us about mitochondrial variation in cardiomyopathy patients.
Great, thank you very much. Sorry for the swap that we had agreed previously, so I'm just going to share my slides and start. Okay. So this will be the focus of the talk, which is mostly based in a recent paper we've published precisely on an analysis of mitochondrial variants from the 100,000 genomes in cardiomyopathy patients. And from those findings, the main result of the paper and focus is this variant M4300, which A to G, which is the cause of hypertrophic cardiomyopathy. So this is basically the paper and my co-authors as well. It's a small research letter type paper. But to start with some background, because I recognize that obviously the audience might be a bit heterogeneous in terms of different backgrounds and so on. So this is in terms of disease, the main focus of the paper, hypertrophic cardiomyopathy, and it has been my main focus in terms of research for many years now. And it's usually defined clinically as hypertrophy of the left ventricle, sometimes affects both ventricles. And with a threshold of 15 millimeters for probants or 13 millimeters for relatives. This is a CMR image of what is called a four-chamber view, showing a very hypertrophied septum in this, if I can show here, in this region. So this is the sort of a very severe sort of textbook example of a heart with this condition. The prevalence is around 1 in 500 to 1 in 200 individuals in the population, so it's quite frequent. Histologically, it is characterized by fibrosis, disarray of the cardiomyocytes, and also microvascular dysfunction. And each of these components leads to very important clinical endpoints, such as heart failure, arrhythmias, including sudden death, for which hypertrophic cardiomyopathy is recognized to be one of the main causes, particularly in young people. The genetic background of the condition was growingly recognized some years ago to be caused by mutations affecting genes that encode sarcomere proteins. So these are the main causal genes, and we can find them in around probably 30 to 40% of patients with hypertrophic cardiomyopathy presenting for the first time to our clinic, with myosin-binding protein C being the more prevalent and followed by beta-myosin heavy chain. Here we have a representation of the sarcomere with each of these proteins in their context. And so these contractile sarcomere genes are the main causes of hypertrophic cardiomyopathy. One of the issues is, and particularly for clinical application of genetics, that as it happens in other conditions, we still have a large percentage of individuals, both with or without family history, which sometimes are more recently addressed as gene elusive or non-familial. That can go up to about 60% of the patients that we test. And then a smaller percentage is caused by so-called gene or phenocopies that can affect other organs aside from the heart. And this includes amyloidosis, Fabry. So some of them are quite rare, others are not so rare. And all sorts of different patterns of inheritance can happen in terms of the causality for those conditions. If it is indeed caused by a sarcomeric variant, usually it is recognized as being characterized by incomplete penetrance, by variable expressivity. So we see very different severity of the condition even in the same family. And in the majority of cases, it's inherited as an autosomal dominant trait. So the possibility of explanations for these genotype negative patients are obviously the transversal problem to any genetic disease of variants of an uncertain significance. So some of these patients are not really genotype negatives, but we just don't know how to interpret the variant that we find in them. Undiscovered causal genes, which keep appearing and with, each of them probably responsible for, each of the new genes is probably responsible for 1 to 2% in terms of causality. Unexplored genomic regions such as deep intronic variation. Lately, patterns of autosomal recessive inheritance were also recognized for some of these genes. And a bit more on the theme of this talk, the possibility of genocopies or phenocopies affecting the heart and causing this unexplained left ventricular hypertrophy. And also another possibility, which is oligogenic or even polygenic inheritance, might explain some of these genotype negative cases. So the reverse angle, which is of interest to this talk, is the possibility of mitochondrial diseases causing cardiomyopathies. Some of them can be quite aggressive. Other phenotypes, aside from hypertrophic cardiomyopathy, can also happen, including dilated cardiomyopathy, restrictive, and others. And some of them might present with an isolated cardiomyopathy without extra cardiac findings, although that is quite an exception, both for mitochondrial DNA, mitochondrial cardiomyopathies, or mitochondrial cardiomyopathies caused by variants in nuclear genes, which are much rarer. So the typical scenario is a cardiomyopathy, many times with conduction disease, which is one of the red flags, but with also red flags of involvement of other organs and systems, including the nervous system. When we see a patient with cardiomyopathy, that should make us suspect of the presence of a cardiomyopathy, but of a cardiomyopathy caused by a mitochondrial variant. But that is not always the case, and this is just a very quick clinical vignette of a 16-year-old previously competitive swimmer that starts having symptoms, mainly breathlessness, diminishing exercise capacity, where we do a panel, an hypertrophic cardiomyopathy panel that is negative, no recognizable family history, some features on the ECG, which are sort of red flags, such as short PR, and these predominant R waves in the right precordial leads. The MRI shows a mild hypertrophy with some scar in the infralateral wall here. And essentially, some of these red flags pointed towards a mitochondrial disease. We tested, and this is the variant that we found, also corroborated by findings on the myocardial biopsy. So the background for this small paper is that more than 50% of cardiomyopathy cases are genotype negatives. Some, like I said, of the mitochondrial DNA, or in most of them, mitochondrial DNA is not tested unless we have red flags. But we know of this variant for some years that is known to cause isolated HCM, but we don't really know what is the prevalence in hypertrophic cardiomyopathy in general, particularly in genotype negative cases. So that was the main objective. This study was nested into my project of reanalyzing whole genome sequencing data from the 100,000 Genomes project to increase diagnostic yield in cardiomyopathy. And particularly for this study, we wanted to identify pathogenic mitochondrial variants and focusing on, like I said, genotype negative cases. So our cohort was a bit more than 1,000 cardiomyopathy genomes. Only a little bit more than 10% of them were previously genetically solved by the usual clinical analysis from the 100,000 genome group that is fed back into the clinical side of things. Focusing on HCM, we had 796 patients with HCM, a little more than 100 of them having a previous diagnosis, so 600 plus without any genetic diagnosis. We used controls matched from the same cohort and in the same sort of number, and also some controls from the UK Biobank that I'll also mention. And so the samples can be seen for those with access to the research environment and are shared in this folder. The methods use the mito-HPC pipeline, for which the reference for the paper is this one here, nested on GitHub. The analysis was mainly done by one of the co-authors, Renata, who is a senior bioinformatician that works with the mitochondrial disease group at Queen's Square. So many thanks to her. We've filtered for variants with more than 10% heteroplasmy. We annotated the variant pathogenicity using MitoMap, and we excluded variants with unrelated phenotypes or low quality of calling. So overall, from those patients that I've mentioned with HCM, we had 4 with this variant without any previous genetic diagnosis and without any other causal variants, including in the usual sarcomeric genes. So this represented 0.6% of genotype negative HCM cases. And the variant was absent as expected in controls or the UK Biobank cohort, and this is what is illustrated here in this figure with a star representing the location of the 4300 variant. Other findings were just some incidental mitochondrial DNA variants, not linked to any cardiomyopathy phenotype and not judged to be overall clinically significant. So only M4300A2G was causal. And were our main considerations that parting from the knowledge that this variant can cause isolated HCM. The MTTI gene where this variant resides is not part of the standard HCM panels. So again, mitochondrial genes will only be tested as part of particular panels in the presence of red flags and so on, which might not be present in the cardiomyopathy caused by this variant. So which relates to this. So the family itself may lack the typical maternal inheritance clues, which is also related to the fact that it is highly variable in terms of penetrance and expressivity, and it has been postulated, such as in other mitochondrial diseases, that variants in nucleogenes might play a role in this, in the expression of this tRNA gene. This is one of the last slides, just showing that in the last iteration from the CleanGen panel that regularly revises the genes causing hypertrophic cardiomyopathy, they do include the MTTI, but not in bold, which means moderate evidence. So this was published more or less at the same time. So maybe if other evidence like ours starts accumulating, this could be upgraded. And so to conclude, we would recommend including MTTI in the HCM gene panels. I think we have evidence enough now, particularly if for genotype elusive or isolated seemingly singletons, with the advantage that the growing use of whole genome sequencing enables this lookup at the mitochondrial DNA, potentially increasing the diagnostic yield, which is quite informative for family screening, of course, and risk assessment in general. So again, thanks to my co-authors, and I will finish here.
Thank you, Lewis. Maybe we should start with this question from Dave. I think he is asking for some clarification about how this specific variant was previously implicated in HCM and how confident you are that it's pathogenic. I mean, have you done any kind of statistical analyses? Because it's clearly very rare.
Mm-hmm.
I mean, it's not present in biobank. Is there actually a significant enrichment in your cohort of patients?
Yeah, so the first papers describing this have maybe 20 years or so, and they do provide all the necessary evidence, including large families and so on, for and some functional data for pathogenicity. So it is a well-known variant. The thing is, we obviously until very recently didn't have the data to explore in an exhaustive way large cohorts of HCM patients with isolated HCM to look for this variant. So it was considered a very rare and still is, but within hypertrophic cardiomyopathy, maybe not as rare as previously thought. And that was one of the main messages.
Right. So it was sort of 4 in, how many patients was it? A couple of thousand?
It's 4 in 600 plus with hypertrophic cardiomyopathy and without a previous genetic diagnosis.
Okay. Dave says, so it should always have been on the HCM panel.
Probably.
And I've got a question too. In Nomad, they recently brought out this map of mitochondrial constraints. Did you look at this residue, this position that's mutated, and look at its level of constraint?
No, we didn't do that analysis.
Be interesting to do that. I mean, given that it's incompletely penetrant, and I guess people might not die of a heart attack until they're after reproductive age, you wouldn't necessarily expect it to be constrained, but it would be interesting to see. I haven't seen anybody use that mitochondrial constraint map yet for kind of improving the identification of pathogenic variants in mitochondria, and I just wonder how useful it is.
Yeah. And that we do have some dramatic cases in pediatric, so very young ages as well.
Okay. Some other questions. What's the heteroplasmy in these patients? Do they have a maternal history of HCM?
So not always. So it can happen, and it's an homoplasmic. It's an homoplasmic variant always.
Okay.
So it's also different from others in that respect.
Right. Are all your patients with HCM SNOMAD encoded in the EHR used at BARTS?
No, no.
I don't quite know why is that an important question, just for the people who are not in this area?
From what SNOMED is, I think, a way of coding diagnosis. I'm also not too familiar with it. I know that it is a coding system used quite frequently, I think mostly in the US, I think. Not aware that it's used in the UK, or at least not at BARTS, as far as I...
Yeah, I thought it was used in GP records in some systems in the UK.
Okay, so maybe...
I don't know why Charles Guttridge is asking this, but it sounds like he has a reason for doing so. Okay, and somebody else says, can you elaborate on the phenotype of all the carriers? Were they similar? Were there any on the syndromic side?
No, so that's one of the interesting things about the variant. They do not have a syndromic presentation. So they present with hypertrophic cardiomyopathy periods with some features that we recognize from the cardiac tests that are typical of cardiac involvement from mitochondrial disease and are, for example, present in MELAS patients with a 3243 mutation. So the cardiac tests are indeed more or less similar to the red flags that we are used to recognize as important to suspect of a mitochondrial etiology. But this particular variant tends to cause a cardiac isolated phenotype.
Okay. And somebody, Manisha Sajnani, is asking, I guess, a general question for the people new to this area. If you are diagnosed with a pathogenic variant in one of these cardiomyopathy genes, to what extent can you prevent these people having sudden heart attacks?
So that's a great part of my job from the clinical side. So basically, so one thing is not everyone not everyone with the diagnosis of with a variant will develop the condition. So we do have, obviously, we screen the patients regularly when they are known to be carriers of the variants and so on and so forth. But once they present with the condition, what we do is, aside from treating symptoms and hopefully give some medication that helps, we very regularly assess what is known as a risk of sudden death, of sudden arrhythmic events in these patients. And when they are judged, so one thing we do is when they surpass a certain threshold of risk, which is basically done with a lot of clinical features and also some from imaging, we can offer an implantable cardioverter defibrillator, so ICD for short.
Right. What's the clinical course, for example, age of onset of the patients with these variants compared to other genetic causes?
So on average, it will be earlier, but we do have, like I was saying, a very, very large spectrum of presentation. So from baby to 30s or 40s with this same variant, and sometimes in the same family.
Okay. All right, maybe we should move on to Isaac's talk now. Am I meant to introduce him? Is that right? Or is Elwyn going to do that?
Yes, please.
Yes, so Isaac has recently finished his PhD here at the Sanger Institute. supervised primarily by Rahela Rabari. I was his second supervisor, and this is a project that he's worked on in collaboration with both our groups plus Aylwin's group. And it was jointly led with Sean Huang, who was a student with Aylwin. And we've just published this, but now Isaac will give you the product summary in 20 minutes.
You're on mute, Isaac.
Sorry. Hello, everyone. Very happy to be here. As Hilary was mentioning, I did this as part of my PhD, and this was recently published in Nature Communications. You can check out our paper by following this QR code in here. So, what are de novo mutations? So de novo mutations are acquired by gametes, are mutations that are acquired by gametes or the progenitor cells, and these have the potential to be inherited to the next generation. These are important because ENMs are a major source of genetic diversity and are a source of sporadic genetic disease, which is genetic disease present in individuals without previous family history of disease. We detect de novo mutations by doing a family tree or exome or genome sequencing by basically detecting variants that are present in the offspring but absent in the parents. So at population level, one of the main factors determining the number of de novo mutations found in an individual is the parental age at conception. This basically meaning that individuals conceived by younger parents are going to have a lower number of de novo mutations, while individuals conceived by older parents are going to have an increased number of de novo mutations. There are some other factors that are known to modify the germline mutation rate. Some of these are chemotherapy exposure prior to conception in the parents, of course, exposure to ionizing radiation, and the presence of ultra-rare homozygous genetic variation in DNA repair genes in the parents again. However, all of these factors cause or modify germline mutation by causing germline hypermutation, which is a rare event in the general population. So in the general population, it's a bit less clear which factors may modify the germline mutation dynamics at population level in a more slight way without causing hypermutation. There is some evidence to think that there are factors that modify the germline mutation rate, and this comes from evidence from single nucleotide polymorphisms, where it's been found that the mutation spectra, this is the types of mutations and the genetic context where they occur, differs across genetically inferred continental ancestry. So basically, when we compare in a pairwise manner, the types of mutation and the genetic context where they occur across different genetically inferred populations, we find significant differences in the proportions of types of mutations and genetic context. Which lead us to think that genetic and or environmental differences, factor differences across populations have shaped the landscape of germline genetic variation throughout human evolutionary history, and leaves open the question on whether these factors are still active in contemporary populations. So in support of genetic factors affecting the germline mutation rate, there was this recent publication showing that common SNPs at minor allele frequencies higher than 1% modified the germline mutation rate of specifically microsatellites, which are repeat sequences in the genome. On the environmental side of this story, tobacco smoking, which is a known mutagen present in the lifestyles of many individuals around the globe, modifies the mutation rate of, again, repeat sequences in the form of mini satellites. Studies in mice suggest that the effect of tobacco smoking is global across the germline mutation, this meaning that it can affect also the germline mutation rate of single nucleotide variation and indels. However, this has not yet been reported in humans or detected in humans. So in this study, we sought to find or study the ancestral genetic and environmental influences on single nucleotide variant germline mutation at scale by using the novo mutations code from 10,000 whole genome sequenced family trios in the 100,000 Genomes Project rare disease branch from Genomics England. So basically, we did three things. We quantified the ancestry-associated differences in de novo mutation counts and de novo mutation spectra. We assessed the influence of genetic factors on the novo mutation rates by estimating the SNP heritability of germline mutation counts for the best represented homogeneous population in this cohort, which corresponds to the European sample, or the European descendant individuals. And we estimated the effects of parental smoking on de novo mutation counts and de novo mutation spectra. So for the first part of this, we subset the full sample of whole genome sequence family trios to keep in trios where both of the parents were predicted to have the same genetic ancestry. The genetic ancestries were based on readily available classifications, which were based on continental level ancestry predictions, which were obtained from projecting principal components from Genomics England individuals on 1,000 genomes principal components and label transferring, basically. So each individual in this cohort was classified as either being of African descendant, American descendant, East Asian, European, or South Asian descendant. So by keeping these trios from whose parents were from the same ancestry, we kept 9,820 trios, from which we generated the novo mutation single nucleotide variant counts, which would allow us to model the novo mutation rate. We classified each of these single nucleotide variants according to their pyrimidine substitution type, in which we were able to classify each single nucleotide variant as either C2A, C2T, C2T occurring in CPG sites, C2G sites, T2As, T2Cs, or T2Gs. And then counted the number of times that each trio had one of these pyrimidine substitutions, and then obtained the proportions of these pyrimidine substitutions per trio. And these we use this to model the novo mutation spectra. We modeled both counts and the spectra using linear regression modeling methods, quasi-Poisson for counts, and multinomial regression modeling for the proportions. We model ancestry as a factor covariates, in which each trio was classified as either African, American, East Asian, European, or South Asian. We included parental ages at conception as for both mom and dad, given that it's known that these parental ages at conception affect the number of mutations that we are able to detect, as well as other technical covariates that could also affect our ability to detect de novo mutations. Just another note on how we are looking at ancestry effects. So ancestry effects are understood in terms of percent-fold changes when comparing ancestry A versus ancestry B across all possible pairwise combination of ancestries. So we are going to be looking at the percent-fold change differences of African compared to Americans, Africans compared to East Asians, and so on. So for the first result, we found a 4% increase on de novo mutation counts in African trios as compared to American, European, and South Asian trios, which equals to around 3 extra de novo mutations for these African trios as a baseline, which would roughly equal to having an European, South Asian, or American father that is 2 years older as compared to African individuals. So we wanted to discard any possible technical artifacts that could be generating these differences across ancestries. So we retested our model while including potential sources of artifacts, in which we included mapping biases, for which we calculated the average number of mismatches for the DNMs found in each trio. We included a measure of potential missed heterozygous variants, for which we calculate the parental variant allele fraction for all the de novo mutations called in each trio. And we also tested whether there were any recruiting biases by calculating the number of potentially deleterious DNMs per trio. And retested our regression models, and basically we observed no change in the effect size or direction as compared to the original model when including the possible sources of artifacts. So regarding ancestral influences on the novo mutation spectra, we found significant pyrimidine substitution proportion differences across ancestries, namely depletion and C2Ts occurring in CPG sites for European as compared to South Asians, and we detected a depletion on C2As for Europeans and as compared to South Asians. So these two differences recapitulate those differences that were observed in using polymorphism data in the heat map that I showed you at the beginning. But however, these are only two of many of those that were detected in polymorphism data. So we tried to find out why, and one of the reasons why is the sample sizes that we have available for testing these pyrimidine substitution proportions. So basically, we randomly subsampled the South Asian subcohort to the n values shown in the y-axis in these plots. And we repeated this 1,000 times to get a mean effect size for the difference of South Asians as compared to Europeans for the pyrimidine substitutions that we found as significantly different between these ancestry pairs. And we observed an absolute 6.58% meaningful change for these two pyrimidine substitutions, only at sample sizes higher than 500 trios, meaning that at least 500 trios are necessary to detect an effect size that is equal or higher than that in polymorphism data for these ancestry and substitution types. Meaning that, why we are missing some of these signals that we were observing in single nucleotide polymorphism data is due to sample size. Another of the possible reasons of why we detect these differences could be due to parental age differences between different ancestries. So it is well known that different populations differ on the time when they decide to conceive a child. And this is also true for the Genomics England data set. So basically here we are looking at mean parental ages at conception differences for paternal age differences at conception and maternal age differences at conception between the same ancestry classes that we were testing originally. So we went ahead and tested whether these parental ages at conception differences were the source of the observed differences. So basically, we bootstrapped an age matching algorithm across ancestry pairs and repeated our associations 100 times on these subsample cohorts. In each round, we kept at least 70% of the original sample size per ancestry while equaling the mean parental age at conception each time, and repeated the association, the linear regression that I described earlier. And we found that across these 100 repetitions of the linear regression modeling in the subsampled and age-matched cohorts, we detected the same effect directions using the same FDR thresholds in at least 90 of the repetitions, which we conclude that parental ages at conception differences across different ancestries are not the source of the observed differences across ancestries for neither pyrimidine substitutions or de novo mutation counts. So next, we quantified the effects of the genetic effects on de novo mutation rate. So for this, we used, rather than trio level de novo mutation counts, we used phase de novo mutations. So basically, when we have trio de novo mutation counts, we can trace back the parent of where that de novo mutation was originated, and we can assign a number of de novo mutations per parent. We kept only European individuals for these tests, given that we are going to be calculating SNP heritability, which requires us to keep a genetically homogeneous population for this. As I was mentioning, we obtained de novo mutation counts per parent, regressed these by different covariates that could be affecting the number of de novo mutations called per parent, including individual sex, given that we partitioned our data set into 3 individual subsets to be able to test the SNP heritability of germline mutation rate for mothers alone, dads alone, and all of the individuals together. We estimated the SNP heritability using the Gremel LDMS methods, keeping SNPs with minor allele frequencies higher than 0.1%. Which we partition it by math and linkage disequilibrium, and which we also assessed altogether. And we included the 21st principal components of common variation for this test as well. We found no genetic influences on the de novo mutation rate, at least for these subcohorts. For none of our tested individual subsets and none of the variant bin partitions, with which we conclude that common genetic variation is unlikely to explain a large proportion of the variance on germline mutation rate in the general population. Next, we quantified the effects of smoking behavior on the de novo mutation rate. For this, we tested again phase de novo mutations counts per parent. And for this, we kept a cohort of 15,732 individuals for which we had access to hospital episode statistic records, or HES for short. These HES records are in the form of ICD-10 codes, which are given by a clinician when they are assessing a patient, so basically each of these individuals. And we basically looked for F17 derived codes, which is the encoding for mental and behavioral disorders due to use of tobacco. And basically, we classified each individual in this subcohort as ever smoked based on the presence of these F17 ICD-10 codes. We performed a linear regression modeling in a similar way as before, including these ever smoked covariates, where we compared smoking versus non-smoking and included the different covariates that I mentioned earlier, testing across 3 subsets, 3 individual subsets, again, mothers alone, dads alone, and all of the individuals together. We found that the smoking behavior is associated with an increased de novo mutation count across all individual bins, with this effect being significant, but being very modest and representing around 1 extra de novo mutation over a lifetime. I don't present a specific slide for or modeling on spectra, but basically we repeated the modeling of spectra while including or encoding for ever smoked, but we couldn't detect any significant differences in the mutation spectra, probably due to the modest effects that smoking behavior confers in the germline. We were then curious to see whether smoking behavior would be causing the differences that we were observing at ancestral level. So for this, I went back to the trio, the novo mutation counts, and kept trios where at least one of the parents had HES data available, and encoded a trio level ever smoked covariates, where in any case where one of the parents didn't have HES records available, that parent was classified as non-smoker. And with this, I encoded trios or a trio level smoking behavior for the parents. And including these covariates, I could not detect any changes in the original associations detected for each of the different ancestries, for which we conclude that smoking behavior differences across different ancestries don't lead the observed differences at the ancestral level. So how about the spectra? So in a similar way as here, I included the parental smoking behavior covariates in the regression, in the multinomial linear regression for detecting epidemic substitution differences, and reproduced the previously detected associations for European and South Asian differences on C2A and CPG to TPG sites. Without finding any additional significant differences. So that plot is too large to be shown in a single slide, but you can check it out in our supplementary material in our paper. So in summary, population level factors, we found that population level factors influence the novel mutation rates. With which we conclude that there are factors actively influencing the germline mutation in contemporary populations, however, with the sources remaining to be determined, which could be either environmental, genetic, or maybe both. We found that genetic factors do not significantly explain variation of germline mutation. And this is very important to specify. This applies for at least the European ancestry, which we tested in here. And this might hint that there is an active selection pressure for removing mutator alleles from the germline, although the presence of these may exist transiently, which has been demonstrated by previous work modeling using SNP data. Finally, we found that tobacco smoking influences the novel mutation rate, and that this has a really modest effect on germline mutation, which equals to around 1 extra de novo mutation over a lifetime. And we didn't find any effect on spectra, likely due to the overall small effect of smoking. And we found that the ancestry effect differences that we detected are independent from smoking behaviors across populations. So that's all from my side. Thank you to Genomics England for allowing us access to this very rich data set. Thank you to the participants for allowing us to research their genomes. To my PhD supervisors, Rahila Rabari, Hilary Martin, and Elwin, people in the Martin group, Rabari group, my co-authors, Senwon Huang, Joanna from Genomics England, and other people that were there for really fruitful discussions. So I'm happy to take any questions.
Great, thanks, Isaac. Let's start with a question. Yes, were trios of each family sequenced in the same run? How much variability of sequencing runs impact detection of DNMs?
No, not all of the trios were sequenced in the same run. Like there were batches of sequencing, as far as I know. Like someone from GEL could specify better. But the whole point is that they do have a really well-established pipeline to avoid differences in regarding technical artifacts due to sequencing in these cohorts. Also, it was tested before in the main work generating this data that differences either in the times or even in the reference genomes used for the mapping they did not affect the number of the novel mutations called per trio. So yeah, it's very short answer is very homogeneous.
Well, and also we controlled, when we were doing all the analyses, we controlled for all these QC covariates, right? To try and get rid of any artifacts of that kind of thing.
Yeah, precisely. So yeah, we are including the necessary technical covariates to prevent these kind of biases or batch effects to contaminate our data. Yeah.
Great. Are there any other questions for Isaac? Not clear how you determine ancestry versus environmental factors. Was there an interaction?
So ancestry is genetically determined. So basically, we use common variation to assign each individual to a specific ancestry, and these ancestries are based on the 1,000 Genomes project. Regarding environmental differences, basically each of these ancestries represents a mixture the effect of each of these ancestors represents a mixture of both genetic variation and environmental effects, but we cannot really dissect this. That's the problem, and that's why this question will remain open for future research. We do not test particularly between any ancestry versus environmental interaction because yeah, it is hard to say like what is the environment for each of these ancestries. Then again, like each of these ancestries is a composite of effects between genetic and environmental factors. Yeah.
And Kitty Sherwood says, if you were to change your assumption that parents without smoking status available are non-smokers, does this change any of your results?
Yeah, we tried to go down and then keeping only trios where both of the parents had his data available and where we would be able to as well have an accurate measure of smoking behavior. But these would remove the effects due to just power because yeah, we wouldn't keep enough trios with complete HES data available. Basically, because also, well, I should have mentioned that the number of trios that we have for non-European ancestry individuals is really reduced. Like the next most common ancestry in our trio data is South Asians, which are only 1,200 trios.
What about a role for epigenetics in this? Someone asks.
Well, I'm not really sure. I'm not really sure if there is epigenetic data available in Genomics England, especially for the trios.
It's a suggestion that due to environmental factors, methylation of DNA might differ between ancestries, and that might change the mutation rate. I think maybe that's what this person is thinking about.
It could
I guess that is definitely plausible, but I guess we can't easily query that because we don't have methylation data in development tissue at scale from different ancestry groups, but that would be very interesting.
Yeah. The fact that we cannot dissect this ancestry or the real culprit for these ancestry associations, like open the door to many questions and many possibilities. One of these could be epigenetics, definitely.
Okay, well, there's 5 minutes till 3, and I think Elwyn's supposed to wrap up. But thank you very much to both of our speakers.
So I will just share one final slide, which I meant to share, I think, which is about the research seminar next time, which is on the 24th of June. And that will be hosted by people from the Therapeutic Innovation and Trials community with speakers Ella Whittle and someone else to be confirmed. So other than that, I think we are concluded today. Thank you very much for everyone very much for coming along, and thank you very much to our two speakers for presenting their research.
And when you receive a survey from us about participation in this research community, please fill it in, because we'd like to know what we can do to help make this more of a community and help you with your research. Thanks, everyone.